So our next speaker is Gerald Tuskan, who's a mainstay of the JGI. He's a distinguished scientist and group leader at the Oak Ridge National Laboratory in Tennessee, and he's had a long-standing affiliation with the JGI, where he's been one of the leadership team of the plant program and all of its instantiations, along with Jeremy Schmutz. Jerry is a key leader not only in the JGI plant genome program, but kind of worldwide, especially within the forest genomics and forest biotechnology world. For his role in that community, he was named the 2012 Forest Biotechnologist of the Year, among the many honors that he's achieved. I've had the pleasure to work with Jerry for the last, I think it's now 10 years. And what's great about working with Jerry for me is that every time I meet him and have a chance to talk to him, I learn something fundamentally new about plant biology, about the agronomy. I often learn things about population genetics, which is a field that I think I know something about already, but talking to Jerry, I always learn something more. And those of you who follow the JGI YouTube channel, stay tuned, because you'll see a great interview with Jerry that we filmed yesterday about the eucalyptus genome, which he won't be talking to us about, but is also one of the many projects that Jerry has taken a leadership role in. This morning is going to kind of blow your mind, I think, with what you can do with thousands of genomes and some clever analysis. Talking about what is your actual topic? Using the rich genomic resources, 1,000 popular genomes, and how that is really revolutionizing our understanding of how wood formation is done in nature, and how we can control it and mold it to our uses for bioenergy purposes.
Thanks, Dan. That was a kind introduction. I've known him for 10 years. He could have been cruel and mean. We've had many experiences together where those stories could have come out, but didn't, so I'm thankful. Also thankful for the invitation to come and speak today. I have the opportunity to stand here and talk about work that's been done or contributed to by dozens and dozens of people. I've listed some of them here, multiple institutions around the world. It's really been an honor to be here at JGI and to work with the staff scientists here and scientists around the world, so it's my pleasure to give the talk today. I'm going to go into the main body of the talk in just a little bit, but I want to give a little bit of background to bioenergy and what are our target goals, at least within the bioenergy centers where we're trying to produce transportation fuels, and then a little bit about cell wall formation and cell wall polymers, in case some of you don't have that background. So our goal is to overcome recalcitrance. And recalcitrance is the ability of a plant cell wall to yield its sugars under microbial conversion. At least that's how we're defining it. And so to do that, to be able to release sugars more efficiently, we really want to understand how cell walls are formed. We know that there are three main polymers: cellulose, hemicellulose, and lignin. Cellulose is deposited initially in the primary cell wall. Hemicellulose, 5 carbon sugars are laid on top of that. And then finally, lignin is deposited. And these are all complex polymers. They're the three most abundant polymers on the planet. And so they have some importance ecologically, but also economically. We're trying to apply genetics and genomics approaches to tease apart how cell walls are formed. Now, the depiction or the cartoon that's a little bit complex here is the presentation of a primary cell wall. And we deal with poplar. Poplar is a long-lived perennial woody crop. And what separates the herbaceous materials from the woody materials is that typically woody materials deposit secondary cell walls. And those secondary cell walls are placed internal to the primary wall. And it really represents the final stages of programmed cell death. So when we see a tree, whether it's a redwood or a poplar or a eucalyptus tree, and we see this tremendous volume of biomass, we're looking at cellulose and we're looking at lignin and we're looking at hemicellulose, but we must keep in mind that most of that material is dead. So xylem has gone through its life cycle, primary walls are formed, cellulose, hemicellulose, and lignin are deposited. Secondary walls are formed, having those same three polymers, and then the cell dies. And what remains alive is the cambium and the apical meristems and the radical meristems. And so if we have an opportunity to modify cell walls, we have to do it at the point when they're formed and when they're first initially dividing and laying or depositing cell wall materials down. Once xylem is formed, once the cell is dead, there's very little opportunity to modify the cell wall. So why are we interested in cellulose, and why are we interested in biofuels? Well, cellulose is made up of glucose. It's a polymer, a long linear chain of glucose. And that glucose can be readily fermented once it's released from the cell wall. And we can do this through microbial or thermochemical conversions. So we take, and this happens to be a eucalyptus plantation in Brazil. I'm going to go back and forth between eucalyptus and poplar here as a way of emphasizing comparative functional genomics as a way of identifying genes that control specific traits we're interested in. So this is eucalyptus. You harvest the chips. You segregate cellulose from lignin. The cellulose can be converted to ethanol, isobutanol, and other forms of transportation fuels. And we're looking at lignin as a source of carbon fiber for spun fibers that ultimately would go into reducing the weight of vehicles, so you'd get improved fuel efficiency. So you can utilize the entire cell wall matrix, the entire components of the cell wall for transportation fuels and lighter weight vehicles. I'm going to focus the talk mostly on lignin. And for those of you that don't know the lignin pathway, this is it here. There's 32 enzymatic steps that start out with phenylalanine, and it goes from acids to aldehydes to alcohols through a series of enzymatic steps and a series of metabolite precursors that ultimately lead to three major building blocks of lignin: guaiacyl, syringel, and H forms of lignin. Lignin is an amorphic, aromatic, hydrophilic, polyphenolic component of the cell wall. And what all this says is that lignin biosynthesis is complicated. It's not a linear chain of glucose molecules. It's actually an amorphic matrix of these three precursors. And how these three precursors are deposited determines how well the cell wall is held together. So if we can shift from G forms of lignin to S forms of lignin, we now know that we can increase sugar release. So one of our targets was, well, let's look at the enzymatic steps that are involved in the transformation of precursors in guaiacyl forms of lignin and move those over to syringel forms of lignin. Seemed very straightforward. A lot of people have done this enzymatically using transformation techniques. So that was our initial thought on this. I don't know if there's anything else there that's important, so we'll skip all of that. So the main body of the talk deals with these questions. Are genes known to affect cell wall biosynthesis conserved across perennial plants, mostly woody plants? Is there a comprehensive parts list that we can then use to design cell walls specifically for our purposes? Can functional comparative genomics help us define that parts list? And once we had that, could we attempt a synthetic biology approach in designing cell walls to yield the products that we're most interested in? It's generally believed now that the first land plants that evolved from aquatic environments had cell walls. And the polymer or the molecule that allowed this to occur was lignin. So at the interface between the aquatic and the terrestrial environment 500 million years ago, the first land plants colonized terrestrial habitats. And these occurred in these tidal pools as relatives of algae in what we would call probably a biomat. And the reason that it's currently felt that cell walls played a key role in that evolutionary development is based on two hypotheses. First is that cell walls give or yield or provide desiccation tolerance to the plant. Without a cell wall, the cell lyses or pops or bursts. And the second was reproductive success. And there's some direct evidence that the first land plants evolved from aquatic plants that produced reproductive structures above the water surface. If you think about competition in an open pollinated environment, anything you can do to favor successful recombination and pollination would give you a selective advantage. And so the reproductive structures popped up above the water surface and allowed successful reproduction. All of those habits, all of those phenotypes required cell walls. So it looks like primary cell walls evolved once, very basally, to all land plants. And this occurred 450 to 500 million years ago. Now, there are a lot of other traits that have subsequently occurred within the phylogenetic tree of life for land plants. We see that pores developed probably once, stomates developed once, vascular tissue, and roots didn't develop until quite a bit later into the evolutionary occurrence of land plants. What I'm going to focus on are the angiosperms, because that's where poplar and eucalyptus occur, and that's where secondary thickening and arboreal forms of growth occurred. So that's where perennial habits are found. I think one thing to note here is that this tree here looks an awful lot like the phytosome page, and that's not accidental. We can take a phylogenetic approach to understanding gene function if we have high-quality reference genomes to make those comparative analyses. And I hope to demonstrate how we've leveraged the resources that JGI has created to do that, to understand cell wall formation. So this little piece of the graph here is now turned 90 degrees, and with a little more detail, these are the angiosperms, the eudicots. It's not important that you read the species that are listed here, but the point is that woody perennial habit and secondary cell wall thickening, or biosynthesis, has been gained and lost many times over evolutionary history. So the original land plants had primary walls. Subsequently, secondary cell walls evolved. And that habit has come and gone many, many times over evolutionary history. So we're looking at about 180 million years of time here. And most common extant crown lineage taxa lineages have both woody and herbaceous members to their groups, phylogenetic clades. So we know that cellulose, hemicellulose, and lignin were present and basally occurred in the first land plants. And when we look at the divergence, the amount of diversity that we see across the angiosperms, across the eudicots, we see that anatomy varies greatly. And there's just some cross-sections of anatomical differences among various woody plant species. But the one thing that's consistent is that the chemistry of the cell walls are conserved. And because the phenomenon of secondary cell wall thickening has come and gone many times, what we're really witnessing here is convergent evolution. So that gave us the first indication that there really is a cell wall parts list, that the plants have used this over and over and over again through speciation and evolution to create the cell walls that we have today. So once we have that parts list, can we put it together in novel ways to create unique cell walls that would then suit purposes in terms of bioenergy, carbon sequestration, carbon fiber production? That's the hypothesis. So Dan mentioned that the eucalyptus genome has the paper has just been accepted, or I don't know if you mentioned, but the genome's been sequenced, assembled, and annotated, and the paper was just recently accepted by Nature, and hopefully it'll be out in print here in the next few weeks. Well, in that process, we were able to take all the high-quality sequenced reference genomes and project the putative ancestral chromosome of all eudicots. So now here's the phylogenetic tree that I just showed you, but these are the genomes that have been sequenced, and most of these have been done by JGI. And you need a high-quality assembly, you need a high-quality annotation to be able to do this type of analysis. From this ancestor, there were 7 chromosomes that went through one whole genome duplication. Each dot represents a whole genome duplication. And over evolutionary time, that ancestral chromosome arrangement was shuffled and doubled, either through whole genome duplications, or segmental duplications, or tandem duplications. So that original parts list that evolved 450 million years ago was captured in these chromosomes, and each of those chromosomes has subsequently been doubled or duplicated or triplicated over evolutionary history. So now the parts list is shuffled in and among the chromosomes of the current land plants. Well, if we look more specifically at eucalyptus and poplar, there's a curious phenomenon where one of the ancestral chromosomes, ancestral chromosome 3, descended to poplar, and now we're talking about 180 million years of evolutionary time, descended to poplar and eucalyptus unrearranged. So there's a high degree of synteny, and this ancestral chromosome was not broken apart and distributed across other chromosomes within either of these genomes. This is the first time that this has been witnessed, and we began looking at this, trying to figure out what genes were on that chromosome that may cause that to be inherited as a unit. So when we did that, this was work done here at JGI by Ina Dubček. This was done using VISTA, and we saw that there were 104 syntenic blocks. This is a dot plot of chromosome 18 on poplar, chromosome 3 on eucalyptus, and you can see the conservation and the synteny between these two organisms that were separated by about 120 million years of evolutionary time. So maybe there is something to this idea of an operon in plants, which we heard about yesterday. Well, when we look at this, there are many genes associated with cell wall biosynthesis, secondary growth in the cell wall. And these genes, there's a tandem array of sucrose synthase 4 that has descended onto chromosome 18 in poplar and chromosome 3 in eucalyptus that had to predate speciation of their last common ancestor. And so that's a characteristic that was common or inherited from the ancestral chromosome state. And that tells us something a little bit about biology. So then we did deep RNA sequencing to look for expression. So I said the original genes have been duplicated through tandem duplications, through segmental duplications, through whole genome duplications. And this is, again, the lignin pathway. You see syringeal guaiacyl forms of lignin, H forms of lignin. This is phenylalanine. You go through acids, aldehydes, alcohols, and these are the enzymatic steps. And because we had a highly informed, highly annotated genome, we were able to look at tissue-specific expression of orthologs and paralogs. And although you can't read it, PAL, for example, has 6 or 7 members, and these are different tissues: young leaves, old leaves, roots, xylem, phloem, flowers, etc. So we could determine which ortholog was expressed in developing xylem. And you paint this across the metabolic pathway, and you begin to have a picture of what members of the parts list were important. And the surprising thing here was that, let me back up and say, before we had assembled genomes, you would have taken a candidate gene, looked for homology in an RNA library, and then assumed that those individuals that had the highest homology, the highest identity, were those genes performing the same function, those enzymes performing the same function. That's not true. What we found out was that the gene family that was expressed in developing xylem was conserved, but the ortholog or co-ortholog or paralog was not. And so you can't use identity to determine which gene is causing cell wall formation, lignin, hemicellulose, or cellulose. So that was the first thing we took away from there. We wanted to know, is this parts list based on the biosynthetic pathways, the entire parts list? So we began to apply GWAS analyses. We assembled a population of 1,100 poplar genotypes collected from across the Pacific Northwest. That's been resequenced. Wendy and Joel here at JGI did all the resequencing and SNP identification. We have 48 million high-quality SNPs. There are many, many others that we've thrown out, but these are high-quality SNPs. There's a SNP every 10 base pair. If you use a minor allele frequency of 5%, there's a SNP every 50 base pair. This is a highly polymorphic organism. Here we're talking about poplar. The SNP assays were then brought into a GWAS, or Genome-wide Association study. We assembled the population. We genotyped the population. We measured replicated phenotypes, mostly cell wall chemistry. We correlated genotypes to phenotypes. We controlled for false discovery rate, and we identified significant relationships based on all of those procedures. When we looked at the enzymatic steps that determined, the terminal enzymatic steps that determined lignin content, cellulose content or hemicellulose content, we never found a significant association. So all our preconceived notions about how to modify cell wall chemistry seems to be contradicted by the GWAS analysis. So chemistry varies a lot, cell wall chemistry varies, but the genes in the terminal pathway are not associated with that variation. We did find significant associations, however. This is work that Wellington Mucharo's done. He's somewhere here in the audience. So this is an example of one of many, many associations. This is a significant association that is significant for both paralogs. So there was a whole genome duplication in Poplar. Both paralogs give a significant association. The two paralogs are 90% identical, and both of them cause a premature stop codon. So a partially functional or non-functional enzyme is correlated with a significant change in lignin. All of the significant GWAS associations with lignin, cellulose, and hemicellulose occur outside of the biosynthetic pathway. And this was a surprise to us. They're occurring way, way upstream. And so you have photosynthesis, fixation of CO2 into glucose, glycolysis entering the Shikimate pathway, and then off of that you shuttle down through aromatic amino acids, flavonoids, tannins, and then ultimately you get down to lignin. So there are many, many biochemical steps from glucose fixation down to lignin. None of the terminal enzymes were associated with variability in lignin. So this is an example of that paralog that I showed that Wellington's discovered. This is a representation of the flow of carbon in a wild-type genotype. So carbon flows through the pathway and ultimately leads to an average of 25% lignin in Populus trichocarpa. When the mutant is present, either one or other of the two paralogs, you get a 50% reduction in lignin, and you get carbon shuttled away from this pathway into fatty acid derivatives. And we had discovered this phenotypically in 2011, reported on it in a PNAS paper, where those genotypes that had this mutation resulted in high sugar release in a natural context. So this we began to wonder, is this a statistical artifact? Do we not have the power to determine associations between those lignin biosynthetic enzymes and changes in lignin content or lignin composition? So we took the resequencing data and approached it in a different statistical manner. We created what are called signature haplotypes. And this is a particular gene. This happens to be the first gene on the first chromosome. It's of no particular relevance to cell wall biosynthesis, but I use it as a cartoon to show you how we did the analysis. So we concatenate all the SNPs within a particular gene, and they're depicted here. We allow for wobble rules. There can be no more than 5 SNP variants, and they have to occur at a certain frequency to be called a signature haplotype. And then we let the resequencing data coalesce into these signature haplotypes. A couple of interesting things on a whole genome that we noticed. One was that there were never more than 20 signature haplotypes for any of the genes. So based on the wobble rules that we applied here, we could compress that 48 million SNP library into a smaller subset of informative concatenated haplotypes, and that there were 909 genes that only had one haplotype. So among the 1,100 genotypes that we looked at, among the 2,200 haplotypes that are potentially available in the population, 909 genes only had a single haplotype. So that tells us that those genes are under heavy selection, and probably no matter what we want to do to modify them, we will be unable to do that. They will probably result in lethal or semi-lethal modifications. So on a whole genome level then, we built all the signature haplotypes, and then we constructed these hierarchical dendrograms. And how we did that was, if you think about a haplotype as depicted here, you can see that there's an evolutionary context about how these mutations occur. So within a population, as Populus trichocarpa repopulated the Pacific Northwest after the last glaciation, there were mutations that occurred, and those mutations occurred locally and then spread locally within the populations. And then through recombination and mating, they distributed into haplotypes of alternate forms. So you can take this structure and recreate a hierarchical dendrogram, which we've done here. This is for a gene on chromosome 10. It's the 14,590 gene model in the genome. And then you reconstruct that statistically to represent it this way. And now, instead of doing SNP by phenotype associations, you do the associations in a hierarchical manner across the dendrogram. So here we have 19 condition states. And you use an analysis of variance approach, and you see, do any of those 19 condition states associate or correlate with lignin, or glucose, or sugar release, or cellulose? And then you can move up the tree and have fewer condition states and fewer condition states and fewer condition states. And when we did that, we found many, many statistically significant associations with glucose release, lignin content, cellulose content. Here's an example of one of those. So this is the consensus. This is the alternate or variant form of this particular gene here. And we found the statistically significant association at this level. So there are 3 haplotypic conditions. These 2 here are represented by those 2 branches of the dendrogram, and this one here. And this particular form gives us a significant increase in sugar release. When we did that across the genome, and we looked at the lignin biosynthetic pathway, we again found no significant associations with any of the terminal enzymes in the lignin pathway. And all the significant associations were for genes outside of the pathway. So we began to think about it. If you're an organism that is in an environment that would require low lignin content, let's say 24% lignin was somehow detrimental and you needed something around 16%, would you spend all the ATPs and consume all the carbon to drive your precursors and metabolic substrates down to the end of the pathway and then terminate the pathway at that point? It seems metabolically and energetically inefficient. So what we think is happening is that in the environment where selection occurs for higher low lignin, higher low S to G ratio, selection is more effective at higher stages in the biochemical pathway. And this seems to hold true across cellulose, lignin, and hemicellulose. So you may have questions. You say, okay, that looks great. There's a reduction in lignin. You see it in the population. But it's just a statistical association. So Tony, a postdoc in our lab, cloned the gene, put it into Arabidopsis. There was no change in stature for the transgenic Arabidopsis. And this again was the poplar mutant. Put it into Arabidopsis, and we see a 50% reduction in lignin with the presence of the single poplar homolog or mutant allele. This is a negative dominant mutation. It's a premature stop codon. We believe that this particular gene has a substrate binding domain that is unaffected, and the catalytic domain, as a result of the truncated message, is no longer available. So what we have is an enzyme that's binding substrate and holding it or withholding it from further modifications in the Shikimate pathway. When we looked at, these are all the mutants in the population of the 1,100. These are the mutants that carry the modified gene. And this is ethanol yield. So now we've gone from sugar release, now we're looking at ethanol yield. And those genotypes that carry this mutant significantly improve ethanol yields under microbial fermentation. 20 to 60% improvement in ethanol yield. So, in conclusion, genes are conserved and functions are conserved across taxa, and they appear to have evolved in a convergent evolution across woody perennial plants. The functional gene families are conserved, but the specific orthologs vary among taxa. The numbers and types of genes affecting cell wall biosynthesis goes beyond the biochemical parts list. And certainly regulatory elements are playing a key role in that. But other novel genes are also controlling where carbon goes and how much lignin, cellulose, and hemicellulose are produced. Most of the natural variants are outside of those terminal biochemical steps. With that, I want to acknowledge the support of DOE through project funding at the Bioenergy Center at Oak Ridge and project funding here at JGI, and some of the collaborators that have contributed to this work. I'll take any questions that you might have.
Other questions for Jerry? Yes. Nav, right? You got to get a right here.
So, Jerry, can you speculate about the low lignin, high fatty acid variant? And
Oh, yeah. Thanks, Jim.
Whether that's which side of it you think is selective, or both, being
Yeah.
selected for, or both?
I showed that carbon was shuttled into the fatty acid pathway. And at the terminal end of that pathway, we end up having these long-chain fatty acids that mimic insect pheromones. And the particular insect pheromone that it mimics is for parasitic wasps. And so it looks like the part of the low lignin is kind of like a sickle cell anemia situation. Parasitic wasps feed on caterpillars that defoliate poplar. It's a very serious problem, foliar defoliation in native environments. So it looks like there's a benefit for carrying the heterozygous condition. So we always see homozygous wild type that have high lignin. And when we have a heterozygous genotype, we get that combination of high fatty acid, low lignin that probably has a selective advantage. And I should mention that in that particular allele, we never see homozygous mutants. They are lethal.
So I'm just curious as to what the implications of low lignin would be to disease resistance of the species.
That's a good question, and I can. So the question was the relationship between low lignin and disease resistance, but I get this question added on: what is the relationship between low lignin and upright form? What's the relationship between low lignin and water relationships? And when we went out to the natural population and we collected these 1,000 individuals, they were of mature trees. And so the genotypes that I showed you on high ethanol yield were from trees that were 80 centimeters in diameter, 100 feet high, probably 100 years old. And so the genotypes in the natural environment that have these genes grow to maturity and appear to have no insect problems, no water transport problems, and no problems growing upright. Then we clonally propagated these into the common gardens. And in the common gardens, these genotypes actually are among the top 10% in terms of biomass production. Under a very drouthy condition at Placerville in California here, as well as under very wet conditions at Corvallis and Clatskanie, Oregon.
Hi. I was wondering if you or your group has been able to model carbon flux through the lignin pathway and put this into some sort of context.
So we did all the carbon flux based on metabolic profiling. I forgot to mention Tim Czaplinski, and he did most of that work. And so we've used metabolic profiling to determine how much carbon is flowing, and then estimating how many ATPs it takes to make those conversions. And there's clearly a savings in the amount of ATP consumed by having lower lignin. And so that metabolic energy then is apparently available for fatty acid metabolism.
So what's the rationale you select or sequence the species from that specific region?
Could you say that just one more time?
You select all the sequence species from that Pacific Northwest region.
Yes.
What's the rationale behind that?
So that's Populus trichocarpa, and Populus trichocarpa is one of the fastest growing temperate species. I probably should have mentioned that at the beginning. So we're interested in poplar in general, And of the species, Populus trichocarpa, Populus tremuloides, Tremula deltoides, trichocarpa occurs naturally in the Pacific Northwest, and it's one of the fastest growing species. So that's why we targeted that population.
Great. Thank you so much, Jerry.